Preparing bacterial smears for staining, microbiology laboratory skills. Most bacteria are colorless, so they generate little contrast in the microscope field. Therefore, to see bacteria under the microscope, it is necessary to apply color using a staining reagent. Once stained, bacteria can be observed and studied in terms of their shape, size, and arrangement. A bacterial smear is a thin layer of bacteria placed on a glass slides and fixed by heat to ensure that they remain attached to the glass. For coloring, it can be prepared from a solid medium or broth. A good smear preparation is the key to a good stain. There are some important things to consider when preparing a smear for staining. 1. The bacteria should be evenly and lightly dispersed. 2. Bacteria should be firmly attached to the slide so that they are not washed out during staining procedures. 3. It is very easy to not know which side of the slide the smear is on. Be sure to label the far edge of the slide. 4. Allow your slide to air dry before you heat set it. Bacteria will boil and cell morphology will be lost. The equipment that we need is glass microscope slides, alcohol burner or lamp cap or Bunsen burner, inoculating loop and needle, glassware marking pencil and lens paper or bibulous paper. And we also need a broth or solid medium culture. While simple to handle, sufficient attention must be given to the preparation. Follow the principles outlined below with precision. Preparation of the glass microscope slide. Clean slides are essential for the preparation of microbial smears. Grease or oil from the fingers on slides must be removed by washing the slides with soap and water, followed by a water rinse and a rinse of 95% alcohol. After cleaning, dry the slides using lens paper. You can also do the rapid passage 2 or 3 times over the flame of the Bunsen burner. And place them on dry lens paper until they are ready for use. Note: remember to hold the clean slides by their edges. Preparation of smear. It is crucial to avoid thick, dense smears. A thick or dense smear occurs when too much of the culture is used in its preparation, which concentrates a large number of cells on the slide. This type of preparation diminishes the amount of light that can pass through and makes it difficult to visualize the morphology of single cells. A good smear is one that, when dried, appears as a thin, whitish layer or film. Different techniques are used depending on whether the smear is made from a broth or solid medium culture. Preparation of the inoculating needle or loop. Depending on the culture medium, a loop or needle is used for removal of the inoculum. Loops are commonly used to obtain a sample from a broth culture. Either instrument can be used to obtain the inoculum from an agar slant culture by carefully touching the surface of the solid medium in an area exhibiting growth so as not to gouge the agar. A straight needle is always used when transferring microorganisms to an agar deep tube from both solid and liquid cultures. Sterilize an inoculating needle or loop by holding it in the hottest portion of the Bunsen burner flame until the wire becomes red hot. The hottest part of a flame is the blue part, which is typically located closest to where the reaction is taking place. This might be the very bottom part of the flame. Once sterilized, the loop is held in the hand and allowed to cool for 10 to 20 seconds. It is never put down. Make a smear from broth culture. Uncap each tube by grasping the first cap with your little finger and the second cap with your next finger and lifting the closure upward. Note: once removed, these caps must be kept in the hand that holds the sterile inoculating loop or needle. The inner aspects of the caps point away from the palm of the hand. The caps must never be placed on the laboratory bench because that would compromise the aseptic procedure. After removing the caps, flame the necks and mouths of the tubes by briefly passing them through the opening of the microincinerator or through the Bunsen burner flame 2 to 3 times rapidly. The sterile transfer instrument is further cooled by touching it to the sterile inside wall of the culture tube before removing a small sample of the inoculum. With a circular movement of the loop, spread the cell suspension into an area approximately the size of a dime. Allow the smear to air dry. Flame the necks of the tubes by rapidly passing them through the flame once. Recap the tubes on the same tubes from which they were removed. Reflame the loop or needle to destroy any remaining organisms. Make a smear from solid medium culture. Place 1 to 2 loopfuls of water on the center of the slide. Open the agar part of solid medium culture. Pick one colony with sterile inoculum loops. Transfer a small amount of the bacterial inoculum from the slant culture into the drop of water. Spread both into a thin area approximately the size of a nickel. Close the cap of solid medium culture. Allow the smear to air dry. Reflame the loop or needle. Heat fixation accomplishes 3 things. To kill the organisms. To make the organisms adhere to the slide. To alter the organisms so that they more readily accept stains, dyes. Slides must be completely dry before heat fixing, or the organism will be boiled and destroyed. If you heat fix too little, the organism will wash off the slide. If you heat fix too much, the organisms may be incinerated. Heat fixation is performed by the rapid passage of the air-dried smear 2 or 3 times over the flame of the Bunsen burner or in front of a micro-incinerator. Labeling of slides. Proper labeling of the slide is essential. The initials of the organism can be written on either end of the slide with a glassware marking pencil on the surface on which the smear is to be made. Ensure that the label does not come into contact with staining reagents. Note that it is important to recognize the side of the glass slide that you put your bacterial sample on. The bacteria smear is ready to stain.